Hello students, I'm Dr. Arpita De. A very warm welcome to my YouTube channel. Today we will be discussing about restriction endonuclease. Here from this picture we get to see two things. One is a scissor and the other is a molecule of DNA. Now how can we link these two things? Restriction endonuclease or restriction enzymes are used to cut the DNA at specific regions. We call them specific sites. It is very difficult to work with a large fragment or a large DNA molecule. So for the convenience of study, restriction endonucleases are used for breaking that large DNA molecule into manageable fragments of manageable sizes, which makes the study procedure easy. These nucleases, they cut the DNA at a particular site and generate fragments which are smaller than the original big-sized molecule. Now, before we go into the topic, we first know what are restriction endonucleases. Restriction endonucleases are enzymes that cleave or cut DNA at internal phosphodiester bonds. These are bacterial enzymes that recognize specific restriction sites that are usually 4 to 8 base pairs and cleave the DNA strands at those sites. This means what? They are very much site specific. Okay, the technology has come into limelight in the early 1970s. All right, it really enabled us to manage huge molecules, large-sized molecules of DNA. The first enzyme was isolated from Haemophilus influenzae in 1970. Okay, now the naming system is very interesting. Here, these enzymes are represented by a 3-letter code. It is written in italics. Okay, HAE for Haemophilus aegypticus and HIN for Haemophilus influenzae. The letter code denotes the origin of enzyme. Now, we should know, why do we call it endonuclease? We call it endonuclease because the cleavage takes place within the DNA molecule. See, it's highlighted here. It is taking place within the DNA molecule, not outside. Okay, now we'll come to the various types of restriction enzymes. There are normally 3 types of restriction endonucleases which we study. These types are denoted with Roman numerals 1, 2, and 3. Fine. Type 1 endonuclease. Type 1 endonuclease has got 3 subunits. The 3 subunits have got 3 dedicated functions. Subunit 1 functions for the restriction, second one modification, and third one specificity. Fine. And to perform its activity, this enzyme needs certain components. What are they? They need ATP hydrolysis for their function, they need magnesium ions, and lastly they need S-adenosylmethionine. The last line of the slide is very important. Please note that it cleaves DNA up to 1000 base pairs away from the recognition site. This is a very important line because for this particular property, this type 1 restriction endonuclease often becomes unsuitable for various recombination studies. Okay, now we will move on to the second type. We'll come to the type 2 restriction endonuclease. In type 2 restriction endonuclease, what happens? We get to see here restriction and modification are performed by two separate enzymes but for the same target sequence. The target sequence is same, but the restriction and the modification, these two functions are delivered by two separate enzymes. Cleavage occurs at specific sites and nowhere else. This is very important. The cleavage is occurring at exact or specific sites and not anywhere and everywhere. Okay? This type of endonuclease does not require ATP hydrolysis, but, well, it needs magnesium ion as a cofactor. Type 2 restriction endonucleases recognize palindromic sequence. Well, what are palindromic sequences? Palindromic sequences are those which are read the same from both the directions, that is from 5' to 3' as well as from 3' to 5'. It's a mirror-like image, you can say. Like the way we spell the word mom, M-O-M. From the beginning, M-O-M. From the end also, M-O-M. This is an example of a palindrome. Okay, like the example that I've quoted over here, G-T-A-A-T-G. When you read it from your right hand side, it reads G-T-A-A-T-G. Again, when you read it from your left hand side, it is again G-T-A-A-T-G. Okay. Now we'll finally move on to the third type. Restriction endonuclease type 3 is one enzyme with two different subunits, one for recognition and modification and the other for cleavage. This cleavage occurs in only one strand. Okay? It requires ATP hydrolysis for its function, magnesium ions, and S-adenosylmethionine. So this is a brief account of the three restriction endonucleases that are generally or that are widely studied. Okay, there is another type for restriction endonuclease that cleave only normal and modified DNA. Okay, recognition sequences are not well defined and cleavage takes place approximately 30 base pair away from one of the sites. Okay, now one question I would like to ask you. These restriction endonucleases, they cleave the DNA. They have got a special property of cleaving the DNA. Now, can you answer one question? How do they protect their own DNA? If they have got an inherent property of cutting the DNA at specific sites, in what way do you think is it not cutting its own DNA? How is it protecting its own DNA? If you have the answer, you can message me in the description box, or else I'll be uploading these answers in a separate video. If this video has helped you in learning something new, please hit the bell icon to subscribe my channel. Thank you for your kind attention.